everyone. Welcome to our afternoon session. We have a few housekeeping items first. First, there's been some questions about the speaker presentations in the share folder. So today's presentations are all now available in that Google Drive folder. So you should have everything you need for today. And tomorrow, they will all be in there by the time the speaker talks. We can give you that guarantee at least. Our speakers like to keep their slides very up to date. And also some questions about the recording. So not the workshops, but the lectures are all being recorded and will be available probably sometime this summer. It will take a while to process, but all the registered attendees will get a notification when those are available. So you'll be able to get them on the same site where you sign up for the course, but you will be notified about that. And finally, we have an attendee who is suffering from a low battery. So if you have an HP laptop power cord that you are not using right now and wouldn't mind letting someone borrow it for a little while, just maybe you could let Samurai in the front know, and we'll get that to the person. So thank you. And then I will introduce our speakers for the next session. This afternoon, we'll be talking about gene and variant curation. Our first session is on gene curation, and that will be split between two speakers. The first is Erin, who is an assistant professor at the Geisinger Autism and Developmental Medicine Institute. It also spends a good portion of her time working on the clinical genome resource. And the second speaker is Marina DeStefano, who is a molecular fellow at Harvard Med School and the Laboratory for Molecular Medicine. So she'll come up halfway through. So let me invite Erin up to the podium.
All right, thank you, Danielle. Like she mentioned, this talk is going to be split into two parts. In the first part, I'm just going to go through with you the ClinGen gene curation scoring method, and then Marina will come in and show you some examples. So don't fret when we're talking about this first part if you're not quite understanding what is meant by certain things. I think that will become clear when Marina shows you the examples. So I have no conflicts to disclose. I actually have the honor here today to be the first ClinGen speaker, so I get to tell you what that is in case you don't already know. So the Clinical Genome Resource, or ClinGen, is an NIH-funded initiative dedicated to identifying clinically relevant genes and variants for use in precision medicine and research. And the way that we do this is that we encourage the sharing of genetic and health data from patients, clinicians, labs, and researchers. We encourage people to share that through publicly available resources such as ClinVar. Once that information is publicly available, we can use it to answer a number of critical curation questions, such as which genes are associated with disease, which variants are actually causative, and is any of this information actionable? Once we have this information, we share it back into the public domain through ClinVar or through our own website, clinicalgenome.org. And all of this is hoping to improve patient care through genomic medicine. So today, we're going to be focusing on this piece here. Is this gene associated with a disease? So the clinical validity of a laboratory test involves a test's ability to consistently and accurately detect or predict the outcome of interest. And when we're talking about a genetic test, we're specifically meaning correctly identifying the causative variant within the appropriate gene. But what is the appropriate gene? How do we know that there is enough information, enough evidence available to say that variation in a given gene causes the disease in question? And why is this important at all? Well, if you're thinking about this in the clinical context, the ACMG AMP sequence variant interpretation guidelines were developed for use in the context of recognized genotype-phenotype associations. And if you think about the pieces of evidence that are in that framework, a lot of them require you to know something about the gene itself, know something about the mechanism of disease, et cetera. And if you can't answer those questions, if you don't know that information, how can you really interpret any variants in those genes? Given this, should genes with little evidence supporting their role in disease be included in multi-gene testing panels, or should those be the ones that you are returning variants in on clinical whole exome or whole genome sequencing? And then if you're thinking about this from the research context, what evidence do we need to see from the clinical standpoint to say that the gene that you identified as maybe a candidate gene in your research is really solidly associated with a particular disease? When can we move a gene from the research space into the clinical space? So with all of that in mind, the ClinGen Gene Curation Working Group created a systematic method to evaluate the clinical validity of gene disease pairs. Through this method, we've defined the criteria needed to assess gene disease validity. We described the strength of evidence supporting that association in a semi-quantitative manner. And this allows users to methodically classify the validity of a given gene disease pair. And of note, this metric was developed in the context of monogenic Mendelian disorders of reasonable penetrance, and it is not intended to replace well-established statistical thresholds used for GWAS or validated methods to define multifactorial disease risk. So these are our gene disease classifications listed here, and I'm not going to go through each one in great detail. If you're interested, these are available in a publication that I believe is in the shared Google Drive that you guys received. We also have a very detailed standard operating procedures document, which should also be in that folder, but it's also available on our website, which is clinicalgenome.org. But basically, gene disease pairs that have evidence, only supporting evidence, can be classified as limited, moderate, strong, or definitive. Definitive is kind of our highest classification, and this means that the gene's role in disease has been repeatedly demonstrated over time. And when I say over time in general, I mean at least 3 years. If a gene disease pair is classified as strong, that may mean that it has considerable evidence supporting it, but perhaps it's been newly discovered and not enough time has elapsed to allow any potential contradictory evidence to emerge. Moderate and limited have less compelling evidence to support them, respectively. We also have a category called no evidence reported. We use this to describe gene disease pairs in which perhaps the gene has been suggested to be implicated in a disease, like maybe it's a member of a gene family in which other genes in that family have been associated with disease, but no variants in humans have been reported yet. And then down here at the bottom, we've got our conflicting evidence categories. We've got disputed and refuted. And this is for gene disease pairs in which there is valid contradictory evidence. And we really encourage you to work with domain-specific experts when you're encountering one of these situations to determine, is the contradictory evidence that is available outweighing any potentially supporting evidence that may be available. One thing to keep in mind with all of these terms is that they are not reflective of the relative risk attributable to any of these conditions. So even if a gene disease pair is classified as definitive, that does not necessarily mean that variation within that gene definitely causes a specific phenotype. So don't confuse those two things. So in order to arrive at one of those classifications, we've got this scoring matrix here. And as part of this, we consider both genetic and experimental evidence. And for our purposes, genetic evidence refers to case-level family segregation or case control data that support the gene disease association, while experimental evidence refers to gene-level functional information that supports that gene disease relationship. We consider replication over time, as I mentioned earlier. This is really the differentiator between our definitive and strong categories. We want to see that there have been at least 2 different publications from independent groups with convincing evidence over at least a 3-year period. And again, that's trying to allow for time for any contradictory evidence to emerge. We do consider contradictory information, but as you'll see as we go through, we don't actually assign any specific negative points to contradictory information. This is really kind of unique to each gene disease pair to say like what would it take in this particular gene or this particular disease to count as contradictory evidence. So we just ask that people going through this framework kind of document it in this space, and then again consider the overall amount of supporting evidence and contradictory evidence when making that decision of whether or not to call this one of the positive classifications or one of the conflicting classifications. So we'll go through each piece of the scoring procedure one by one, but we'll start with our genetic evidence scoring. And one thing I'd like to point out is that we do provide a default score for many of the categories, and that's just kind of what we think is usually appropriate to assign for a piece of evidence in that category. But there's also a range, and that's just to give people flexibility. We understand that not every piece of evidence is the same as every other piece of evidence. Some things are stronger than others. So if you're in a situation where things are more compelling, you can feel free to upgrade. And if you're in a situation where things are less compelling, you can feel free to downgrade. So we'll start with the case level data section. Each variant can be given points for variant evidence, so in the context of the appropriate mode of inheritance, as well as segregation if applicable. And again, this is to kind of going back to that flexibility that I was just talking about, to allow things that have more information, more compelling information, to get more points within the matrix than something with less compelling information. So for example, if you've got a loss of function variant that is segregating with disease in a large family, you might want to assign that more points than you would with a missense variant with no other family history information. As part of the scoring of case level variant information, we do consider the variants that have been reported. And this is kind of a chicken and an egg scenario. So at the beginning, I told you, if you're not really sure if the gene is associated with the disease, it's hard to interpret the variants using the ACMG-AMP sequence variant guidelines. But variants that have been reported are still valid information that contributes to the strength of evidence for your gene disease pair. So when you're looking at these variants, you're not necessarily able to do a full ACMG-AMP sequence variant interpretation, but you just need to make sure that these variants are plausible, that they have at least some kind of information that would suggest that they could be disease-causing. You don't want to be counting variants that are clearly benign, like perhaps if they are very frequent in the population, we wouldn't want to count them here. Things to consider for non-loss of function variants, typically missense. We do like to see that there has been at least some impact to gene function. And again, like I said, the threshold for this could be low, but you just need to make sure that the variant is at least plausible. And your scoring should reflect that. So if you, say, only have computational predictors that suggest that this variant could be positive, you would downgrade from that default score that's here. Typically, we'll do 0.1 points for that instead of 0.5. But if you had more compelling functional evidence, you could feel free to upgrade. When you're dealing with loss of function variants, you could consider upgrading if the disease mechanism appears to be loss of function and you see that the gene is fully constrained for loss of function variants and you know that it's a severe onset pediatric condition. And you might consider downgrading if you are seeing things like alternative splicing or the variant is near the C terminus and or nonsense mediated decay is not predicted. You should also just in general consider the statistical expectation of de novo variation in the gene in question as well. For segregation evidence can be used to support a gene-disease relationship, though it must be considered carefully because, of course, segregation implicates a locus and not necessarily a particular gene. So when we're looking at segregation information that we can find in the literature, for example, we always consider the methods that were used to identify the candidate variants, and you can see that there's some different scoring, whether the gene was identified by candidate gene sequencing versus exome or genome sequencing, or if they've looked at all the genes in a candidate region. And we have detailed guidance for scoring based on LOD scores for reasonably penetrant Mendelian disorders in that SOP document that I mentioned at the beginning, which should be in your Google Drive folder. We do also consider case control data if it's available. One difference here you'll notice is that we only have a range provided of 0 to 6 points. We don't have that same kind of default that we had for the case level evidence. And this is because, as you know, case control studies, the quality can vary widely. So we really want people to pay careful attention to the quality of a particular study and score accordingly. So things to think about include things such as variant detection methodology, the power of the study, whether or not there are any issues with bias or confounding, and what was reported for the statistical significance within the study. The other large scoring area that we have in this matrix is experimental evidence. And we have set up the categories for this evidence type to be consistent with the MacArthur et al. 2014 paper talking about interpreting sequence variants. And so briefly, our categories are biochemical function, protein interactions, expression, functional alteration, model systems, and rescue. And kind of as outlined in that previous paper, we've decided to weight these things a bit differently. So you'll see that there are lower scores associated with the functional category, so biochemical function, protein interaction, expression, some higher scores within the functional alteration, and the highest scores within the animal model and the rescue section. You'll note that each category has a maximum score that's associated with it, because we felt like we didn't want something to get the maximum amount of experimental points, say from this kind of less compelling piece of information like function, this particular category. You'll see that you could score up to 8 points here, but we cap this at 6, so that no one piece of experimental evidence, so that if you're in a situation where perhaps there's not a mouse model or there is no phenotypic rescue, some of these higher scoring experimental evidence pieces that you can have, that you can still reach the high score with enough compelling evidence from other categories. Again, with the contradictory evidence, as I mentioned earlier, it's not quantified in the summary matrix. Manual review and expert input is needed to accurately assess this type of information. We don't give, like I said, any particular negative points for contradictory evidence, because we really want you to take in the situation as a whole and say, given what I have that's contradicting, is this significantly outweighing what I have in my supporting category? Once you have all of this information together, again, we come back to the scoring matrix. You add all these things up to help you come to a calculated classification using the ranges here. And one thing that I will say is these points are simply intended to be a guide. When I see people doing this, sometimes I see people really fretting about the exact number of points to give to any one thing. But just to keep in mind that the end result is actually these classifications, not necessarily the numerical score that you get. So while the points are good in that they move you towards a certain direction, the end goal is not the specific numerical points, it's the category. And that being said, at the end of the day, if you've tallied up all of the evidence that you found and you feel that given what you've learned, what you think the classification should be is not what it's coming out to be, you absolutely can opt to change the classification. That's why you see a difference here, curator classification versus final. We just ask that if you do this, you provide a very clear rationale for why you've done that. And we've certainly seen that within our various ClinGen curation groups, where the experts have looked at the information and felt that a different classification may be warranted based on current understanding of that particular gene disease pair. So how might you use this type of information? So, kind of going back to what we talked about in the beginning, for clinical laboratories, they could consider using something like this in their test development. So maybe if they are creating new multi-gene panels, they can consider only including genes above a certain evidence threshold. Perhaps even in their result reporting, perhaps they would consider only reporting variants in genes above a certain evidence threshold. Perhaps for clinicians, when they're ordering tests, they may want to consider maybe only panels with established genes, because as you might imagine, if a panel is overloaded with these genes of uncertain significance, you are going to get a lot more VUS back, and that's potentially confusing for both the clinician and the patient. So where can you find this information, or where can you find more information about this process? This is a screenshot from our website. It's clinicalgenome.org. Right here at the top, there's a button called Learn About ClinGen Curation Activities, and there you can learn about our various curation activities, as it says. But there is a button there for gene disease validity, and it'll take you to a page that looks like this. Here we've got a copy of the paper, the various scoring matrices. Here we've got that very detailed standard operating procedures document, as well as some training materials that you might find helpful. And then where do you actually go to find the results? I'll just back up one. You can click on this button here to see a list of all of our gene disease curations that we've done. Or alternatively, you can search for any gene you like up in this top search bar. If you were to click on that button to see all of the gene disease curations, you'd come to a list that looks like this. Here you can quickly see what the classification is and the date. And if you were to click on that, you would see a summary matrix kind of filled in with the various PubMed IDs that were used to back up each piece of evidence and how each thing was scored. And you can also see the ultimate classification. And coming soon, we will have a more detailed view of this information with more text kind of explaining exactly what from each publication made us score something a certain way. With that, I would just like to acknowledge the following people, and then Marina is going to take you through some examples using the framework that we just went over. Are there any just general questions about the framework before Marina gets up? Oh, yes. Oh, and could you come to the mic, please, for the recording?
Maybe just repeat it quick. It's a quick question. Are you doing allele associations or gene association with disease?
So this is just for monogenic Mendelian disorders of moderate penetrance, nothing to do with GWAS.
The question is, are you associating an allele with the disease or a gene with the disease?
Well,
you imagine some genes have multiple different diseases.
Yes, absolutely. So if a gene has multiple different diseases, we could curate all of those diseases. Right now, we're focusing on gene disease pairs that are associated with our various clinical domain working groups. So it could be that a given gene has 5 conditions associated with it, and 2 of them aren't associated with one of our given groups, and so we might not get to those right now. But we do plan to try to make sure each disease for a gene is curated.